In a dihybrid cross, there are two traits crossed at the same time. Unlike monohybrid, mono means one, so we are crossing just one trait. But for a dihybrid, two traits. In reality, there are thousands of traits that are crossed at the same time. But for simplicity's sake, we are going to be just crossing two, and whatever mechanics of how traits are passed into traits will still be the same if there are three, four, five, though the genotypic combinations of the alleles of the offspring will increase. So for our context in our problem, we'll be using two diseases of the two systems we have tackled. The rheumatic fever, the disease of the cardiovascular system, and the COPD, the disease of the respiratory system. So before we do the problem, let us get to know a little about these two diseases. So, rheumatic fever is a result when a person who has strep throat, and strep throat is caused by a specific kind of bacteria, Streptococcus pyogenes, infecting the throat area. So that's strep throat. But once this infection is not treated, it will result to high fever, and if it's prolonged and it will reoccur every now and then, the heart will already be affected, and this disease is now called RHD. There are some people who are susceptible to RHD because of two factors. If the environment is unclean, no proper hygiene, then Streptococcus pyogenes can infect your body and you'll be more susceptible to it. But there are also multiple studies that genes can also play a factor in how susceptible you are for the infection of Streptococcus pyogenes. Now, these genes are not directly related to RHD, but they somehow play an of important role on how the body is affected and how specifically the heart is affected, and that leads to RHD. So the two genes that are mentioned here is HLA gene, or human leukocyte antibody gene, in chromosome number 6, and IgHV, immunoglobulin heavy chain gene, in chromosome number 14. These two genes are collectively called antibody genes because they they code for a substance called antibodies and stimulates the production of leukocytes or white blood cells. The antibodies and leukocytes are important because if there is infection of the body, these are the cells that attack them. So whenever a person gets sick, the first line of defense is to increase the body temperature to kill the bacteria. So fever is not itself a disease, but fever is an indication that your body is fighting the bacteria or virus. The next thing the body is going to do is to produce more white blood cells and antibodies that would further help destroy this bacteria. Now, if the person's genes are healthy, a certain amount of antibodies and leukocytes are going to be produced enough to to help the body kill those invaders. But if these genes are mutated, there will be overproduction of antibodies and white blood cell. And once this overproduction of these cells, they can attack the valves of the heart. So to make this clear, though, Streptococcus pyogenes does not affect the heart, but Streptococcus pyogenes stimulates the body to produce these antibodies. If that person has a defective gene, then they are susceptible to RHD because, again, overstimulation or overproduction can lead to scarring or wounding the valves of the heart. Now, valves are very important because it's just like the doors of a house, it opens and closes. So the valves open and closes to prevent backward flow. Our blood circulates in one direction. So in our case, in the right side of the heart, the blood, deoxygenated blood, enters the right atrium and moves down to the right ventricle. To prevent blood from going back to the right atrium, this valve closes. And once the heart contracts, it pushes the blood towards the lungs, and there is a valve here that prevents the backward flow of blood to the right ventricle. So it ensures that the blood really goes to the lungs. Once the blood from the lungs go back to the heart, it enters the left atrium, and then a valve opens. The blood is inside the left ventricle. The valve closes, preventing the backward flow. And this blood that is here in the left ventricle is pushed out of the heart towards the different parts of the body. And no blood comes back because there is another valve that prevents the backward flow. For those who have RHD, their valves cannot fully close. Hence, trickles of blood can move backward, and it will lead to different kinds of symptoms. So, fever alone can be treated with medicine, but for those who have RHD, it needs surgery to repair the damaged valve. If no surgery is undertaken by that person, then it will lead to the person's eventual death. So genes uh, is one, or multiple genes are one of the factors why there are some people who are susceptible to RHD. Good thing about this disease is its inheritance. It follows recessive. So the dominant one is having no RHD, and for a person to manifest this condition, that person must receive two recessive alleles to show in the phenotype. Now, it's the same with COPD. In some rare cases only, genetics plays a role. It's inherited in a recessive manner. And COPD is a collection of lung-related disease like asthma, emphysema, bronchitis, pneumonia. And it appears that this person who has COPD has an underlying disease which is called AATD. So AATD is known as alpha 1 antitrypsin. This is a protein which neutralizes the effect of neutrophil elastase, which is produced by the liver. Now, neutrophil elastase is a substance that can destroy the cells of the liver as well as the lungs. So, to prevent neutrophil elastase to attack the cells of the lungs and heart, alpha 1 antitrypsin neutralizes it. Just like, for example, you add acid and water, it neutralizes the acid. So it's the same manner here. It neutralizes the brutal effects of neutrophil elastase to the tissues of the lungs and the liver. However, since there is since there is a mutation of that gene that codes for alpha antitrypsin, the mutation leads to decrease or low amounts of alpha 1 antitrypsin, and neutrophil elastase can do its attack. This is just a rare case, but most COPD cases are caused by smoking. It's not just risk, it's one and the only risk of persons, even though without gene defect, can develop COPD because they are smokers. Some will say, I'm not a smoker, but I have COPD. Another reason to that is you might be a secondhand smoker, wherein you are around persons who smoke, and mm, that can still cause you to develop COPD. Let's trace how these two traits now can be transmitted as one crossing. So it states here that both parents, both meaning mother and father, are heterozygous without RHD and COPD. So both parents are healthy, but when we say heterozygous, they have the parents have the recessive allele. In short, the parents are carriers of the disease. But since there is a dominant trait, they do not exhibit or they do not have the disease RHD and COPD. So what is the chance of their offspring to be just like them, carriers? And what is the chance that the gene mutation will be inherited to the next several generations in their family line? In our problem, we'll be using the letter B to represent the allele for with or without rheumatic heart disease, and Y for with or without COPD. Step 1, we write the allelic symbol. So the allelic symbol for no RHD is capital B, since no RHD is the dominant trait. So with RHD, takes the smaller letter of the dominant trait, which is now small b. This is one trait. The next is no COPD is a dominant trait, which we will use capital Y to represent it. And the recessive with COPD will take the small y because it's the recessive trait. Second trait. Step 2, we we assign or will write the genotypic symbol of the parent's traits. In the problem, it states that both parents are heterozygous without RHD and COPD. Heterozygous, so capital B, hetero, small b. Heterozygous COPD, capital Y and small y. It could also be read as heterozygous without RHD, and we use and heterozygous CO without COPD. First trait and second trait. So let's assume this is the mother. So the father's trait is also the same, and it has the same genotypic symbol. Okay, so at this point we have to remember that the two traits must be passed on. So they must appear in the gametes, the egg cell and the sperm cell of the parents. Both must appear in the gametes. To do that and to simulate that, we'll use a method used by your math to multiply terms, which we call the FOIL method. So FOIL is an acronym for first, outer, inner, last. So we'll start with the mother, heterozygous without RHD and COPD, and we'll do the first, the outside part. So there is a chance that this allele representing without RHD will pair up with this allele representing without COPD. And these two alleles can be found and carried by an egg cell. There is also a chance that this allele signifying no RHD will pair up with an allele signifying with COPD. So here is an egg cell that has this possibility. Let's go for the N. There is also a chance that this allele symbolizing with RHD pairs up with an allele symbolizing without COPD in the egg cell. And there's also a chance that this allele and this allele will pair up and they become the set of genes found in this egg cell. Okay? So the gamete, or what we call the egg cell, carries two different alleles. And these two different allele codes for the two different traits. This one and this one. So we'll do the same with the father side. We'll also still use the FOIL method. Okay. First, so there's a chance that the egg cell, the sperm cell, will carry both the dominant alleles without RHD and COPD. There is also a chance that the sperm cell is going to be carrying this one and this combination. And we'll have the inner, another egg cell has this combination of alleles, and also this one. Okay. So again, if you check the gamete, which means the sperm cell, carries two different alleles. One that codes for RHD, and the other one codes for COPD, two traits. In dihybrid, we form four possible combinations: four from the mother and four from the father. And this is supported by the law of segregation. Allele of the trait of one trait separates and combines with the other alleles of the other traits, now forming the genome of the individual. Okay, so step 4, we do the placing of the gametes carrying the allelic symbols of the two traits in their proper places. Okay, so here, since there are four possible combinations from each of the parent, we'll have a 4 by 4 Punnett square resulting to 16 possible combinations. So if we take the percentage, you'll just have to do a little math. Each box is around 6%. That's 100 divided by 16. Okay? So that's step 4. Now step 5 is we combine their gametes because that's how new organisms are formed. And to visualize the alleles they bring, so it's here. So there is a chance that this egg cell will be fertilized by this sperm cell with a very specific allele combination. Fertilization happens, new organism is formed, and the new organism has this combination. Now take note, I did not place there BY, though it can be correct. However, it will be just confusing when we are going to interpret. So better we'll just cluster the same trait, the same allele representation. So it's easier for us to interpret. Okay. You will know the logic as we go to step 6. So let's continue. There's a chance that this egg cell will be fertilized by this sperm cell. Okay. You can predict now what will be the genotype of the offspring. Okay. Okay. So there is a chance that this egg cell will be fertilized by this sperm cell. Okay. There is also a chance that this, so excited, uh, fertilizing the egg cell. And the possible genetic combination. Let's do the rest. And it's the same idea. They are chances of which egg cell and sperm cell carrying the different allelic symbols signifying the traits that, that are combined. Okay. Sorry. As And we're done. Okay, this is step 5. So let's proceed to step 6. Step 6 is um, interpreting, but of the table that you have seen, from the table that you have seen, it's clearly showing the law of independent assortment that the allele from the female parent and allele from the male parent pair or assort randomly, okay, regardless of the other traits. And it results to offsprings showing no specific pattern of inheritance. So in other words, this allele separates with this allele, and this allele separates with this allele, and they segregate in a different manner, in a random manner. So we'll now interpret by calculating the percentages of the phenotype and genotype and indicating their ratio. So we'll do this by column or by row. It's up to you as long as all the 16 genotypes are interpreted. So let's start with the genotypes. Okay, so here is the first genotype. So both are capital letters. So we'll say homozygous without RHD and homozygous without COPD. Okay, so I'll place 1 here to signify that I already have one genotype like this. The next table is homozygous without RHD and heterozygous without COPD. Okay. Next, we'll have this. How about you do it on your own or be with me? So we have heterozygous without RHD and homozygous without COPD. Okay, so we have one. The next box is this. This is heterozygous without RHD and heterozygous without COPD. Next, I'll place one. Next column, this is homozygous without RHD and heterozygous without COPD. And I think this one and this one the same. Okay, so this one. So I'll just have it plus, sorry. Okay, homozygous. So this is plus 1. Next is homozygous without RHD and homozygous with COPD. Okay, that's 1. Next, we have heterozygous without RHD and heterozygous without COPD. And I think they're the same heterozygous. Okay, here. Next, we have heterozygous without RHD and homozygous with our COPD. Uh, I think we need a new one, and it's 1. Next, we have heterozygous without RHD and homozygous without COPD. Where? I think this one. Okay, this one. Next, we have heterozygotes without RHD and heterozygotes without COPD. And I think, okay, this one and this one. So heterozygous. Okay, there. Next, we have homozygous with RHD and homozygous without. So we need a new one. And that's one. Next, we have here homozygous with RHD and heterozygous without COPD. Okay, so we have one, a different genotype. Next, heterozygous without RHD and heterozygous without COPD. Okay, I think, okay, this one works. Next, we have heterozygous with RHD, without RHD, and homozygous with COPD. I think we had it previously. Hmm. Okay, here. So heterozygous. Okay, this one. Next, that's homozygous with RHD and heterozygous without COPD. Okay, so this one. And last is homozygous with RHD and homozygous with COPD. Okay, so here we need a new one, and we have interpreted already the 16 and we have already the tally. So our genotypic ratio is now 1 is to 2 is to 2 is to 4 is to 1 is to 2 is to 1 is to 2 is to 1. So you just follow your tally for the order of your GR. Now it's time for the phenotype. So I'll be using uh, my data here for the phenotype. Okay, so the first phenotype of is this homozygous without RHD and COP. So dominant without. So the person will have without RHD and COPD. I'll tally it as one. This one is still without RHD and without COPD still. So there are two. I'll add it plus 2. Now here, without RHD still and without COPD still. Okay, so I'll add plus 2. And still without RHD and COPD. And this is 4. So I'll place here 4. Now this one is homozygous without RHD and without COPD. So without without RHD and with COPD. So different phenotype. Okay, both of the this one is both without. This one is different. So I'll tally 1. This one, heterozygous without RHD. So this is without RHD. And homozygous with COPD. So without RHD, but with COPD. So plus 2. Okay. Next, homozygous with RHD and homozygous without. With with RHD and without COPD. Okay, and that's one. Next, this one is homozygous with RHD and without COPD. So this is plus 2. And the last is homozygous with RHD and with COPD. Okay, 1. Therefore, our phenotypic ratio is 9 is to 3 is to 3 is to 1. If you have the percentage, you'll just have it, um, if you say this is around 6%, so 9 times 6, you have the percentage now. Okay, so this is step 6. Now we're ready to answer step 7. And just to recall, okay, it's time for us to answer the problem or the question. And this is the last step in answering the dihybrid cross problem that we had. The question is there a chance that the offspring will be like his or her parents who are heterozygous without RHD and COPD? For the correct answer, the answer is yes. It can be seen from the Punnett square that the combination of heterozygous without RHD and COPD, or capital B and small b, capital Y and small y, is possible. The Punnett square shows the possible genotypes of the offspring basing from the allele combination of the parents. Furthermore, the Mendelian laws explain the possibility of having a genotype of heterozygous without RHD and COPD, namely the law of segregation, where one allele of one trait segregate and become part of the set of genes in an egg or sperm. Law of independent assortment, where one trait do not depend on other traits on how they are assorted. And last, the law of dominance, where dominant trait will cover a recessive trait in heterozygotes. For contextual justification, as we can see from the context that it has 25% chance of forming offspring having a genotype of heterozygous without RHD and COPD. How did we get 25%? So all we have to do is divide 100 by 16. So each box in the Punnett square is 6.25%. And in the tally of the genotype, heterozygous without RHD and COPD has 4. So 6.25% times 4 is 25%. Thus, in another way to interpret, their offspring of this couple has a 25% chance. So it's a less than 50% chance or 75% chance, but there is still a chance of a heterozygote offspring. Last question, is there a chance that the gene mutation causing RHD and COPD will be inherited to the next several generations in their family line? Well, there are two possible answers for this, depending on which genotype are you looking at. Yes, if both parents have the recessive form of the gene mutation, or no, if the dominant alleles are present in the parent. For the scientific principle, the law of dominance states that the dominant allele masks or covers the recessive allele, while the laws of segregation and independent assortment state that alleles separate and mixed with other alleles randomly in order to form a unique set of genome of the individual. For contextual justification, if the answer is yes, then as we can see from the context that it has a 6% chance of a genotype BBYY if the parents are both heterozygous. And if their child carries this and meets another person with the same genotype and produce an offspring, the gene mutation can be passed on. If the answer is no, then it's couples who have both homozygous without COPD and RHD in their genotype, then the mutated gene cannot be passed on. Or you can have couples who have these genotypes. The genotypes are not both homozygous dominant, they are considered carriers, can pass on the mutated gene or genes, but unexpressed in the phenotype of their offspring. So I hope everything is clear in solving the dihybrid cross. Thank you for listening.